Now we'll continue with the applications of cell culture. So what is the answer for the poll just now? Cell culture can be used as a tool in molecular and cellular biology studies. The answer is yes. So in fact, cell culture can be used as a basic model system to look into specific cell activities or behavior. For example, transcription, expression, in terms of how cells interact with each other. So we can also look at the genes expressions or proteins expression, the cell signaling, as well as cell proliferation, cell migration, or even cell adhesion. So cells can also be used for toxicity testing. So it's particularly useful for predicting the adverse effects of some chemicals or drugs. So it gives a predictive acute toxicity profile using the cell cultures. So in this case, you can use the liver cultures or the kidney-derived cell cultures because they have a role in the metabolisms of chemicals. So other than that, cell culture can be also used in the applications of tissue engineering, for example, generating tissue constructs or scaffolds. So for example, like the patients with burns, okay, so you will need to culture a piece of skin tissue before it can be transplanted back into the patient with severe burns. So other than that, we can also look at the propagation and differentiation of embryonic stem cells and the adult stem cells using the tissue engineering technique. So cells can also be used for cancer research. So in this case, we can compare the differences between normal cells and cancer cells in terms of their genes and proteins expression. Okay, so we can also manipulate the cultured cells to express certain oncogenes, for example, and look at what are the effects of these oncogenes onto the cells. Do they proliferate in a very rapid manner, or does it induce cell death? So we can also use cell lines or cultured cells to identify suitable drugs or treatments that selectively kills these cancer cell lines. Apart from that, it can also be used to produce vaccine, okay, because like for example, virus, you will need to use cells to culture viruses. So virus, they are obligate parasites. That's why they are cultured using cell lines to produce the virus. So we can also use cell cultures to detect or isolate viruses and to investigate how virus actually infect the cells. So cell cultures can be also used to produce pharmaceutical products, for example, proteins like monoclonal antibodies or hormones. So in this case, cells are used as a host to produce the proteins of interest. Other than that, it can also be used for genetic counseling. Okay, so they can detect abnormalities of the chromosomes in the fetal cells. So we'll go through the historical development of how cell cultures um, developed throughout the years. So culture of cells begins in the 1900s, where they culture primary explants of tissues. So these primary explants are actually samples of tissues or organs from other organisms. So scientists that start culturing these explants, which are derived from tissues from other animals, okay, and try to culture them in lab. So in the 1950s, they start to use trypsin. Okay, so what is the function of trypsin? Trypsin is actually a protease enzyme. So it can be used to dissociate the tissues into single cells. So in this case, the use of trypsin actually helps to establish cell lines. Okay, because cell lines, they are single cells that can be attached in a two-dimensional layer. Also in the 1950s, they have developed defined medium for the culture of cells. Prior to this, cell cultures, they are using biological fluids or serum from other mammals in order to support their growth. So with the development of the defined media, it can be used to culture cells in a more consistent way. So in the 1970s, they start to introduce the cell fusion techniques or genetic manipulation. They want to know what if we combine two cells together and form a hybrid of the two cells, what will happen to the cells? Does it induce tumor, or does it induce genomic instability or reprogramming of the cell genomes? And then slowly they start to culture other different cell types, like for example, mesenchymal stem cells to be used for the tissue engineered cartilage. So this is mainly for the tissue engineering or the repair system. So in the year 2000, they have developed the Human Genome Project. So they start to look into specific genes or proteins or genetic deficiencies from this human genome. And in year 2007, they have a breakthrough in which they are able to reprogram the adult cells to become stem cells. So this is the one that we have talked about in the first two slides about what are induced pluripotent stem cells. So actually, they are adult cells that are reprogrammed back to the stem cells state. So in year 2008, they also found that these stem cells can be reprogrammed by using valproic acid. So valproic acid is actually a mood stabilizing drug. It can be used to treat seizure or epilepsy. So valproic acid is a histone deacetylase inhibitor. So they found that valproic acid can be used for direct reprogramming and to generate the induced pluripotent stem cells.